So hello everyone in the what we are reading video blog of the South American Mycorrhizal Research Network. I am Cesar Marin, once again with an interview. This time around, we are not asking questions specifically about some article or some paper. Instead of that, here we have to Dr. Caroline Gutjahr. I'm hoping I'm pronouncing your name correctly. She is currently a professor in the Technical Munich University, but we have heard that she will start a new position as a director of a Max Planck department. So, hi Caroline, many thanks for being here, and tell us first, which is your current situation, your current job situation and research situation, and then what is this new position about, and when do you start, and what are your occupations in this new exciting position?
Thank you very much, Cesar, for the invitation, first of all, to speak here and to talk to you in this blog. And just as a small anecdote, because you were talking about my surname, it means actually Gutjahr in English, or buen año in Spanish, or American community. So I'm currently indeed a professor at the Technical University of Munich. I've been here since September 2017. And recently I got a position as a Max Planck director at the Max Planck Institute of Molecular Plant Physiology in Potsdam-Golm, which is close to Berlin. So now I'm in the southeast of Germany, and I will move to the northeast of Germany when I take up this position. And the current situation is that I'm still employed at the Technical University of Munich, but as a side job, I already slowly start as a Max Planck director, so that I can already start organizing things a little bit at the Max Planck Institute before we completely move with the whole research group to the new institute. And we will move there most likely in February 2023 and start the new department. And you were asking what the new position is about. I think so. The difference is that as a Max Planck director, you have basically you are leading a whole department, and together with other directors at the same institute, you are leading a whole institute. And this is of course bigger than a professorship at a university. And it will give me the opportunity to also hire some group leaders that can have a kickstart into their career in my department. And of course, my dream would be that we will be a nice community there with complementary expertise, so that we can bring together different expertise, for example, in biochemistry, molecular biology, and cell biology, and all work together our understanding of this very exciting symbiosis. And another great opportunity is that there is basically at the Max Planck Institutes, you have long-term secured funding. So I don't need to write a research proposal to get a grant for every PhD student I want to hire. Yeah, so I can write grants, but I don't have to. And this gives you, of course, a little bit more freedom and more opportunities to also start more risky projects, because you don't need to think about the next grant all the time.
Oh, that's fantastic. So I guess that brings me to the second question, which is, you mentioned a lot of, like, I think, interdisciplinary work, and the question will be, will you have like a main focus, like a main research agenda, or will be different kinds of topics in arbuscular mycorrhizal fungi?
So there will be an agenda, but of course there's also the possibility that the agenda will change over time, because you realize that after 5-10 years moving into one direction, that now another very exciting direction comes up that you want to follow. Or sometimes you make an unexpected discovery, which will bring you into a new direction. And so my current agenda is to, for the moment, continue on the path that we have already started. And this is on one hand to understand how quantity of colonization is regulated. So what are the molecular factors and how do they work to regulate whether a root is colonized at a low level or very strongly. Yeah. And in this respect, we work on the karrikin signaling pathway, which is a novel hormone signaling pathway for which the receptor is known, but the hormone is actually not yet known. That's very exciting, I think. And this pathway was discovered in the context of post-fire germination in Australia. So it was found that there is this compound that is produced in smoke of burning vegetation. It's a small butanolide, and this is called karrikin. And then in the second step, the laboratory of Stephen Smith, together with Dave Nelson and Mark Waters, who are now PIs themselves, they found the receptor for this compound in Arabidopsis. And then it turned out during my PhD that this receptor in rice is also required for Arbuscular mycorrhiza. And we are now trying to understand what are the downstream signaling mechanisms that downstream of this receptor, which regulate the quantity of mycorrhiza. And of course, it is also very interesting to find out what is actually the ligand of this receptor, which is, of course, in the context of mycorrhiza, not karrikin, because there's no smoke and no fire involved. Then we have a second research line. In this, actually, in the first research line, we also care about how environmental conditions influence colonization. For example, we have recently published a paper on how the phosphate status of the plant regulates the amount of colonization, and we will extend this to more nutrients and also to look at other environmental factors. And then we are interested in how the formation of arbuscles is regulated from the plant side, because you know that this is a very interesting process. The plant cell is remodeled to be able to take up the arbuscle or to host the arbuscle inside. And here we mostly focus on transcriptional networks. And I think we still have quite a lot to do to decipher these networks. But we also are interested in cellular mechanisms that basically drive this cell remodeling, and in nutrient exchange between the two organisms. Because as you know, there is this periarbuscular membrane that surrounds the arbuscle, which is plant-derived, and this hosts a number of nutrient transporters. And we think that many of them may not yet be known, and we would like to look into this. And then as a third research line, we look at so-called functional compatibility. And this refers to the observation that it depends on the plant-fungal genotype combination, how much the plant profits from the fungus. We call this also mycorrhiza responsiveness. Yeah, how much is the growth increase in low nutrient soil when you inoculate a plant with arbuscular mycorrhizal fungi. And this differs between lines of the same species, for example, between different maize lines. And we would like to use these differences to perform QTL mapping to find genetic polymorphisms, which determine the growth difference between colonized and non-colonized plants. And then in the future, I would like to also look a bit more at the fungus, because we have mainly focused on the plant side so far. And I would like to understand better what are the responses of the fungus, for example, to root exudates, and also to see whether we can make this fungus genetically tractable in the future, which is currently not the case.
Oh, that's a super succinct list of the research lines that you will develop. It seems to me that is very broad. I like the part where you mentioned like on the focus on the environmental factors, and also to have the fungal perspective, because obviously this is always missing in many works. So that's super exciting. I think that my last question is a more practical one, and is how and I guess that is the same procedure for all Max Planck institutions, but how future students, postdocs, researchers can associate, can join to this exciting department that will be ready next year.
Of course, I would like to say I'm of course very much looking forward to motivated and interested PhD students and postdocs. And there are several ways to join. First, of course, just write me an email with your CV attached and describing your research interest. This is very important. Several people don't do this. Yeah, they just write an email and say, I'm interested in your research. I would like to join your lab, but they don't even say that they have understood what my research is about. Yeah, so it will be important if you want to get my attention, because I receive lots of emails, that you describe why you like mycorrhiza, what you find especially interesting about it, and what is the reason for contacting especially me and not someone else. Yeah, and so that is the first entry point. And then there are different ways. Either you are lucky and I have money and can support you right away, or you can apply for your own funding. This is especially possible for postdocs who can apply for postdoc fellowships. But also for PhD students, there are funds from the German government, for example, from the German Academic Exchange Service called DAAD, or some governments give fellowships to PhD students to go abroad. For example, China is doing that, and I believe also some South American countries do that, as far as I know.
Yeah.
And that is a possibility. And then for PhD students, there are also two ways. Either you contact me directly, or you apply through the International Max Planck Research School, which is a special graduate school at the Max Planck Institutes. And here you have every year a deadline where you can apply, and then you go through a general interview process. And this allows you not only to apply to one lab, but you could also have a look at other labs and see, maybe during the interview you realize, oh, this was not as interesting as I thought. Maybe I want to do something different. Yeah, and then you could also of course, you need to be accepted by the principal investigator of that lab. Yeah, but there is also still the possibility to see other laboratories as well.
And
does this answer your question?
Yeah, totally. I was thinking, for example, maybe people who want to do research stays or go and visit for some months, or propose projects together, either with German or European funding or funding from elsewhere. I think that also can be a possibility, right?
Yes, absolutely. If it fits, so it must fit into what we do in our lab. Otherwise, it doesn't make sense. Yeah, I also sometimes get requests. For example, now my professorship is called plant genetics. And of course, plant genetics is a very broad field. And then I sometimes get requests. I'm working on the genetics of XYZ, and therefore I would like to join your lab. But it has nothing to do with what we do in our lab. And of course, then it is impossible to accommodate someone, and it also doesn't make sense. Yeah, but if someone can really profit from what we do in our lab for their research, because it fits very well, then we are open to host guests.
Excellent. Thank you. Thank you very much for a very nice interview. Really excited to hear about this. Really excited to see how mycorrhiza and how specifically AMF is going so big to the point of having a Max Planck department. So that's great to hear. And I don't know if you want to say a final word to our audience.
Yes, I can basically only agree with you. I'm also very excited that we got this chance and that Arbuscular mycorrhiza is getting this big opportunity. I mean, there is already a lot going on nowadays in the world, but this will definitely add strongly to the progress in Arbuscular mycorrhiza research. And as I said, I'm looking forward to exciting applications as well. Excellent. And thank you very much for the for the opportunity to talk. And also, I hope we maybe meet sometime in person at the conference, for example.
I hope so, yes. Well, this year ICOM is mostly online, but yeah, I hope so.
Yes.
Okay, thank you. Goodbye.
Thank you. Bye.